Okay, welcome everybody to FORGENOM II project webinar number 3 of 2025. And today we're going to have a seminar by Dr. Victor Epain. He's currently a postdoc researcher in computational biology in the BIAS lab here in Milan University at Milano Bicocca. And he did his PhD at the INRIA Glen lab at the James Scale team. His work is mostly about genome assembly as scaffolding and implemented graph algorithm and graph structure to formalize these problems using mostly mathematical programming, which is his most expertise. And today he will do some mathematical programming in plasmids. And the title of this seminar is A Journey into Mathematical Modeling for the Plasmid Binding Problem on Assembly Graphs. So thank you very much, Victor, for this webinar, and the stage is completely yours.
Thank you a lot, Simone, for the presentation. So yes, my slides are titled A Journey into Mathematical Modeling. Why a journey? Because we have biological problems and we want to address these problems through, for example, optimization, combinatorial optimization problems. So we have first to define them. It's by itself a problem to solve, defining the problem we want to solve, and then to propose some algorithms, some strategy to solve the defined problem. And obviously we want some tractable approaches. So a journey into mathematical modeling, in which context the plasmid binning problem and on a certain type of data, assembly graphs. So what I will present to you is a joint work with Mattias Gros, Gianluca Della Vedova, Paola Bonizzoni from Università degli Studi di Milano Bicocca. And I also worked alongside Cédric Chauve and Annick Epain from Simon Fraser University in Vancouver, and did some works with my former colleague and friend Kérien Thuillier at University Paris-Saclay. So thank you for the opportunity you gave to me to present this work at FORGENOM II. So before we start, some questions, but okay, I will go directly to the introduction. So what is plasmid binning? What is its purpose in the sense of why we want to solve the binning problem, for which biological purpose, for which further biological analysis, and how this plasmid binning problem has been addressed in the literature. So first, the plasmid binning is in the process of studying antimicrobial resistance, so for clinical problematics issues. Plasmids are short extra-chromosomal, so they are alongside the chromosomal DNA sequences of bacteria. They are in bacteria cells, and they are mobile genetic elements, which means that they can move either through different bacteria or incorporate the DNA sequence of the bacteria. And when bacteria exchange plasmids through horizontal gene transfer, they can exchange then antimicrobial resistant genes, and this is problematic in the clinical field, of course, because some bacteria which were not able to resist in a certain environment can, through these exchanges, resist and provoke some new diseases or new pandemic. So the plasmid binning is a stepping stone process towards antimicrobial resistant gene exchanges analysis. And we can define the pipeline process as follows. We first want to classify some DNA sequences. So we have a bunch of DNA sequences in a metagenomic sample, for example. And we have both DNA sequences from the chromosomal part of the bacteria, from the plasmids, from the viruses. We want to classify them such that we say yes or no, a sequence is part of a plasmid. Then we want to bin them, and we want to type them in detail from the class. So you have, imagine you have short reads, you sequence bacteria samples, so you have a mix of a lot of organisms. You assemble them, you obtain contigs. You want to first classify these contigs. So in green, we say that, okay, these are candidates to be plasmid sequences. Then you want to bin them. Binning is differing the plasmid sequences into different groups we call bins. And then we want to type them. The typing is approximately a step to label the bins to do further analysis. So which type of analysis we want to do. So the typing, so labeling the bins we have obtained from the classifying results. Typing helps to, for example, study if an antibiotic resistance epidemic is driven by one dominant or several plasmid types, and can enable to reconstruct phylogenetic relationships based, for example, on core gene SNPs. So a label, what is the definition of a label? It's a type, what we call a typing scheme. A typing scheme can be based on a replicon, so it's a backbone loci for plasmid replication, and you have the mob typing schemes, which is a backbone loci for mobility functions, for plasmid mobility functions. However, typing schemes, so it's useful for study antibiotic resistance epidemic, for example, but it's still providing limited resolution for epidemiological inference. Today we will focus on binning. You have many tools that follow this way of structure the binning pipeline, let's say, with some tools dedicated to each step. But we will focus on the binning problem. So binning problem, from a classification result, you want to bin the different contigs into different groups. Some of the tools are using assembly graphs, so contigs with links. Some of them just compare sequences between a database. It's the case of MobRecon, for example, from the MobSuite pipeline, which compare sequences with the replicon and relaxases database, so specific sequences of plasmid already known, and then made a complete linkage clustering based on match distance. Plasmid spades is based on spades, which is a De Bruijn graph assembler, and search for circularity because plasmids are mainly circular, and try to merge the contigs with really high coverage. Plasmids are more present, the plasmid sequences are more present than the chromosomal sequences. You have also plasbin flow. I will go further into the details of this method because all my works were based on plasbin flow. It's a mixed integer linear programming flow-based formulation on assembly graphs using some features on the contigs. And you have also G+CC working on assembly graphs based on the statistical seed and extend walks strategy. Refined then. Okay. So how to define the plasmid binning problem? So you have let's say you have an assembly graph. You have some contigs with some features. These features can be coverage, a plasmidness score. So more you are high plasmidness score, more likely you are a plasmid sequence. You have also links between the contigs. That's why we have an assembly graph. And you have some biological knowledge, like the plasmids are circular, mainly. The output we want is a set of bins, which is groups of contigs, with the following properties. A contig can belong to several bins. And we do not bin all the contigs. Some contigs are chromosomal contigs. So we don't want to bin them into plasmid bins. So because I'm a computer scientist, my next question is what computational or algorithmic problem I want to solve. I have to solve. Is it a hard clustering problem? Hard clustering is I have n groups, unknown number or known number of groups, and I want to put each sequence in exactly one group. The answer is no, because one config can participate into several bins. Is it fuzzy clustering? Fuzzy clustering enables to say, okay, one sequence can participate at 0.5 score to this group and 0.5 score to the other group. It's fuzzy and not really, because in the fuzzy clustering, you have to give the number of clusters. And here we don't know the number of plasmids we are reconstructing. Sorry. You have an adaptation of the fuzzy clustering, the robust learning fuzzy CMINS. Okay, it's a fuzzy clustering approach, and this approach is interesting because is independent in the number of bins. So perhaps we can use this. Yes, but we have contigs connected by links, and this type of framework is not considering graph structures. So we should go further into dedicated graph binning problem approaches. And so a journey into mathematical modeling. I will base my presentation on plasmid flow, which is an iterative flow-based plasmid binning approach on assembly graphs. It has been published in 2023 by Anik Epain, Masra Fezranemoun, Thomas Vinar, Brona Brejodva, and Cedric Shaw. And I based the following work to a new hierarchical multiflow plasmid binning approach. And the goal is to robustly defining the binning problem on assembly graphs and implementing a tractable approach. So first I have to present in some details a plasmid flow, how it works. So it works on assembly graphs. You have contigs, you have links, you have contig coverages, you have contig plasmidness score. Okay, you can obtain them from a classification result. You have also, in plasmid flow, they want to label. It's not really typing, but they still label the bins, and they label with the GC content of the bin globally. And so they cut the GC contents ratio into different intervals of GC content, so big K interval. And for each of these intervals, each config is associated with the GC content probability to belong to this GC content interval. Then you have a set of seed contigs. The seed contigs are contigs identified as plasmids with a really high plasmidness score, for example. What they output is a list of bins, and each bin is described by a list of contigs, okay, associated with their normalized multiplicity, and each bin is labeled by the GC content interval. Okay. In the figure in the right, I just remind you that a config can participate into several bins and also participate, not in the figure, but can also participate to the chromosomal, let's say, set. Okay. Plasmid flow is based on assembly graphs and is an iterative flow binning approach. I haven't defined yet the flow approach. I will first define what is iterative approach. In the iterative approach, let's say I haven't drawn the links, but each point here are contigs, and you have your assembly graphs without the links here, and you have the desired bins, 3 bins here, blue, orange, and red. Okay, what you begin with the entire assembly graph, and you don't know anything. Okay, you begin to find a first bin. Then you extract the bin, and you update the coverages because you use a certain amount of coverages for the contigs, and so you have to update the coverages in the graph. You continue, you find the second bin, you find the third bin, and all is perfect. You loop on step 2, so finding a bin and extracting it from the graph until no seed is remaining, and so you ensure that each bin has at least one seed. Okay, so plasmid flow is an iterative approach based on a flow problem. So I don't know if I have to describe what is a flow problem, but let's say a flow problem is based on a network. And as an example, you can imagine the water network under a city. And the idea is, okay, the pipe are limited in the flow they can receive and they can send. And the idea is, I will send some water in the network, and I hope the water will go properly into the network. Okay, we will use this idea of flow network, but we will not use water, we will use the coverages of the contigs. And so we are not in a real configuration of a flow problem here. We have first an assembly graph, and we want to transform it to a network flow. In order to, we apply a flow problem. Okay, so an assembly graph here, I'm giving you two ways of representing an assembly graph. The first way is an artistic view, and it's really convenient because you see an arrow represents the orientation of a config because the sequencing is double-stranded. You don't know the strand of a config, of a read, and then of a config, so you have to consider the two strands, and so you obtain this arrow. If you follow the arrow in the right sense, it's forward orientation. So you take the sequence of the config and you don't change it. If you take the reverse, you take what we call the reverse complement, or the reverse complement of the sequence of the config. It's not a graph, it's an artistic view. It's really cool, but in a graph, you obtain an undirected graph, but it's not really convenient in mathematical. So I will use the graph illustrated in C, which is really explicit because we duplicate according to the orientations, and we obtain directly arcs, which are directed edges. And so we can transform this graph directly to a network flow. So how to transform to a network flow? You need a source and a sink, or a target. I will use sink. The source here is connected to only the seeds, and each vertices is connected to the sink. We need a capacity, a limit on the flow we can put onto the arcs. And here the arc capacity is the minimum coverage between those of the two contigs involved in the arc. You cannot use more than the minimum one of the two contigs involved in the relationship between two contigs. Okay. You cannot invent the presence of a contig. It's another way to say that. And okay, so now we have our network flow. And what is really convenient with mathematical programming is you can express yourself according to properties you want to ensure for a user. So from the point of view of a bioinformatician who want to apply a problem, you have many ways to solve combinatorial optimization problems because every paper sometimes present their own definition of the problem. And there are some advantages, there are some inconveniences. You have to know the properties of the results you get in order to do further analysis, correct the analysis, etc., etc. So here, what are the properties of the results? First, you take a bin, all the contigs are connected together through paths in the graph. Okay, they are not connected directly by links all together. They are connected. The bins considered all together, so you take all the bins, and you must ensure that you are not using more than the coverages of each contig, of course. You cannot invent the presence of more present of a contig. Each bin contains at least one contig seed. You have length to consider a bin, and they associate each bin with a normalization coverage coefficient, which represents a proxy of the plasmid multiplicity in the bacterial sample. Okay. About the quality of the bins. They ensure that the minimum quantity coverage of a bin is high. Why? Because the plasmids are more present than the bacterial sequences in the samples. So we prefer to have high coverages. Almost all the contigs GC contents respect the label of the bin, which is the GC interval. Almost. Why? Because you have some short contigs that for which the GC content ratio is not in the interval, but it's not so important because almost all the contigs respect this interval. And almost all the contigs in the bin have a high plasmidness score. You don't want to, because the plasmidness score testimony if a contig is more likely to be a plasmid sequence or not. Okay. I will perhaps take some time to explain what is a meal formulation because we have the property, okay? We can say some properties, but how can we ensure them through algorithmic processes? Mixed integer linear programming is a really efficient tool to ensure some properties until you can write them linearly. Because it's mixed integer linear programming, you have to model like a game. The game you have to define some rules, you have to define some objectives. The rules and the objectives must use only linear expressions. So linear expression is variables multiplied by coefficient plus or minus coefficients multiplied variables. For a rule, you can use inequalities, and the objective function is just a linear expression. You want to maximize, for example, so maximize the score, or you want to minimize if you want to minimize a cost. In practice, you define your mathematical optimization problem, of course, from the real world. Now you hope you can write a linear constraint to represent it. So you write a mixed integer linear programming model. So you write the variables, you define the rules, and you define the goal. Then you send to this magic black box, which is a solver. Like you can use open source solvers or commercial solvers with academic license, for example. Then you retrieve the solution given by the solver and you interpret it into your real world. Okay. So I will not go further into these details. It was just like to illustrate how technically they ensure some properties without giving some math because it's not really useful. If you want to go further into the details, you can make stop in the video here. Okay, perhaps here it's quite important because I gave you some bin properties. Okay, I also gave you some quality of the bins, but it's not so easy to ensure these properties. And here we can just model the things with one objective. And so all these qualities are merged together. And how the authors of Plasmin Flow managed that? They wanted to maximize a score. Let's say the plasmidness score, something like that. They want to maximize what? The total flow. The total flow corresponds to the maximum coverages you are using for your bin. Sorry, the minimum coverage you are using for the bin, of course. And so, of course, if you want to maximize the total flow, you are maximizing the use of the coverage. And the plasmids are more present than the chromosomes, so you are using really high coverage. Okay. Almost all the contig GC content respect the GC interval. You have a GC term, which acts like a penalty for active contigs whose GC content differs significantly from the overall bin GC content from the label of the bin, which is the GC content interval. Okay, so they describe it as not linear, but you can use some tricky things to make the way it's not linear. You have a coefficient, the GC penalty, for the contig C corresponding to the GC content interval B, then they multiply by the binary choice of choosing or not the config, and the binary choice of choosing or not the interval for the bin. And so they multiply two binary variables. This is not linear, but you can use some tricky strategical things to transform it to linear constraints. Okay, linear variables and linear constraints. The final quality, almost all the configs in a bin have a high plasmidness score. This is quite easy to write because you have the term GD, which acts like a penalty, either or a reward, either if the configs have a low or a high plasmidness. Okay. This is a whole property-based formulation. And now I have presented you plasmin flow. I want to go toward, with a S, a robust multi-flow binning approach. Perhaps there is no S sometimes. Okay. Whatever. The question I will address here are which plasmin flow weaknesses can we address and can we solve? Because sometimes we are limited by the technical choices we have made, especially with the MILP. We want to keep the iterative flow approach in the first time. How to adapt the flow approach, keeping this context, okay? Then we will see that it's not sufficient. And I will answer the question, why should we define the binning flow approach as a multi-flow problem? And then, because we are in a mixed integer linear programming and it's already time costly, I will address the question, difficult question, is the multi-flow approach tractable? So I have told you that plasmin flow ensures the connectivity of the bin. This is true, but through heuristics. And we wanted first to incorporate the connectivity constraints of the flow directly in the MILP. Because they first find a bin, they verify it is connected. If it's not, they redo the experiment. So we want to not redo the experiment and directly have a connected bin. So I work with Karen Trillier. We have a preprint on some SAT framework to ensure a connected flow. So we call them SAT variants of the connected flow problems. Okay. Second time, which is not a performance issue, but more a quality issue. In plasmin flow, remind that when they transform the assembly graph to a network flow, they add a source vertex, they only connect to the seeds. And in the circularity of plasmids configuration, you can have some problems. Let's take the first example. In the first example, you have ABC normally connected through circularity. But imagine that there was a problem during the assembly process and there is a missing link. And here CA is a missing link. So what will happen? The solver will first take B because you have to go from the source to a seed because the source is only connecting the seeds. You take B and then you take C and you cannot take A because there is a missing link. So the dash are the elements not taken by the solution. The solver can also take BA in reverse, but not C. If the circularity is maintained, it's not a problem because you can find BCA or either BAC. Okay. And I want to show you that the solver will be biased according to this circularity artifact. Imagine that A, the solver really wants to take A together with B and C. And if the benefit is great, perhaps the solver will pass through another config, let's say D, which can be a config from another plasmid, to connect through paths B, C, and A. And this is a problem because you are using another config. You can lose in recall for another plasmid, or you can lose in precision in your current bin. So what is the solution of this problem? It's a problem of adapting the data, actually, not the modeling by itself. We can consider that adapting the data is part of the modelization. The solution is to connect the source to all the vertices, of course. And a question we can ask is, okay, you are adding more variables, and so you will add more constraints. But okay, you are adding more variables, and it will be more costly in time. This is not so true, because the solver, when you incorporate this type of weird behavior in A, the solver will have to choose between B and C or B and A. And you are creating an artifact of choice in the branch and bound of the solver. The branch and bound is what? The solver is a black box, but even without knowing too much details about the solver, we can say that the idea of this solver approach is to have this tree of possibilities. And the strategical approach is not to explore all the tree of possibility, but to choose which branch of the tree, when I have to make a choice, which branch of the tree of the possibilities I will take. And the branch and bound is using bound to decide if it's a good idea to explore a branch or not. You have a lower bound of what you can expect of the objective function, and you have an upper bound. And if these two bounds are the same, it means that you reach the optimality and you stop. And in that case, you are playing with the bounds and you product artifacts, and so the solver doesn't really know which type. I mean, it is possible that the solver cannot really know which branch he should take. While when you had a lot of, you add all the source arcs to all the vertices, the solver will not take a lot of time to choose, because he will say, okay, I can directly take A with B with C, and I will not choose between B and C and B and A. So, okay, you have added more variables, but you help the branch and bound. Okay, it's a tricky thing. But the worst problem of Plasmid flow is not this type of previous issues, but more in the iterative approach, binning approach. And you have what I can call the enlarged first bins bias. What this means? So imagine you have your assembly graph. You first find the first bin. If you remind, these bins you have found here is a merge of two plasmids. Why? Because it is possible that in this type of configuration, so be careful, the colors are the same, we can say, but the number of contigs not. Sorry for that. But imagine you have two plasmids, P1 and P2, and they are connected through non-plasmid contigs. You want to maximize the score. What will happen in that case? It is possible that the solver prefers to merge P1 and P2 because, okay, he will take the non-plasmid contig, it will be a penalty, but the penalty is smaller than the benefits of merging the two. And so you have the enlarged first bins bias. Why in plural? Because you have this history of searching bins after bins. So the first bins are have this benefit. But it's not a benefit for us because we are merging two plasmids in this case. And so you obtain a bin with two plasmids. Then you search for another bin. Okay. And then the two plasmids are merged, so you have a loss of precision. You can have mixed effect. You can have also a loss of recall, because if the solver is taking a contig from another plasmid to merge two plasmids, perhaps this previous contig that helps to merge the two plasmids will not be used after that. And so it will miss to the next plasmid. So you can either lose in precision, but either in recall. And you have combined effects really difficult to apprehend and to debug. So we must redefine the plasmid binning problem. So what is really the goal? The goal is to find a set of unknown number of plasmids bits. That explains the best our data. Our data can be coverages, plasmidness, seeds, and I will not take the GC because the GC is part of a labeling, and for many reasons I don't really want to use the GC because of clarity. You have to reconsider the new sequence formed by the concatenation of the sequences and linearly, because the probability of the GC is not linear. You cannot correct linearly the GC probability if you concatenate two sequences. So it's an heuristic. Okay, I prefer to explain the data I can already explain. So we will search for several bins in ounce. So from the iterative flow searching approach, we will go to a multi-flow solving approach. Then we will use biological knowledge in the graph structure. We know that almost, plasmids are mainly circular. Also true, but okay, let's consider circular plasmids. We will search for them. We will search for them with seeds, and some of them not necessarily with seeds, because some plasmids cannot be reconstructed if you miss the seed. Okay, but it's more confident to search for plasmid with seeds first. Okay, what are the bin qualities we want to ensure? We want to encourage or either penalize the coverage usage for contigs with either a high or a low plasmidness score. We want also to penalize the unused coverage for contigs likely to be part of plasmids. We want to trust the classification result. The classification results gave us plasmidness score. So we want to use this data. And we will trust the previous tool. Okay, we want to also be robust according to the contig length. And in part of globally whole binning quality, we will adopt a parsimony approach. The fewer bins we need to explain our data, the better. Okay. We want to search for circular plasmids, but some links may be missing from the graph. So we will search for circular, complete circular plasmids, and either partially circular. And the method I'm presenting to you is a multi-flow binning approach, yes, but also hierarchical. Hierarchical, why? Because we want to search for circular plasmids with seeds, then circular without obligation of seeds, then partially circular, blah, blah, blah. Okay. The multi-flow binning approach. So what will happen comparing to the iterative approach? First, begin with the entire assembly graph. Then we first find one bin. You will say, okay, you fall into the same problems as before. Yes, let's continue. I'm saying now I want to search for two bins. Now the solver will likely provide me the two different plasmids. Why? Because the objective function is better to not take the non-plasmidic contig. Right? Now I'm allowing a new flow such that it corresponds to a new bin. I'm searching for 3 bins, then I'm searching for 4 bins, but here the solver, so I force the solver to find exactly 4 bins. And it will do his best, and his best here is splitting a bin in 2, splitting a plasmid in 2, and we can put constraints that ensure we have a better explanation score, a better objective function, and we can add a cost of, if you really want to add a new bin, you will pay a minimum cost, and you have to be better than this minimum cost, plus the previous objective function, of course. If you are not better, you are explaining the same data, but with more bins, or you are explaining badly the data, let's say. Here it is the case, so we keep just 3 bins, and we extract them. We update the coverages in the graph, and we continue the multi-flow binning approach. We loop until no seed is remaining, until we reach a certain threshold of number of bins we want without seeds, etc. Okay. Okay, the difficult question is, okay, again, mixed integer linear programming is really costly. So you are not searching for just one flow, but you are searching for multiple flow, and the unknown number of beans, for example, can be upper bounded by the number of seeds, and this number of seeds can be really high. I mean, imagine just 10 flows on an assembly graph with 100 contigs and 300 links. You multiply by 10 flow, so you have to multiply 10 times the variables of one flow, etc., etc. So it can be really costly. How to make it tractable? You can use a practical and strategic approach. First, you model the problem for all the beans. Then you activate one bean per one bean. So you say, search for exactly one bean, then search for exactly two beans, etc. Gurobi, which is a commercial solver with free academic lessons, I found already good in solving mixed integer linear programming models, keeps in cache the explored branch and bound tree. Remind this tree of possibilities. It will keep cache. So I want to benefit from the caches in order to, when I will add a bin, I want to Gurobi update. Again, it's a black box, but I can trigger Gurobi. And I want to use the cache Gurobi preserved to make the second solve with 2 beans and with 3 beans, etc. faster. Gurobi can ensure the reuse of the cache if I'm modifying the constant part of the inequalities of the constraints, etc. And we can do that in our context. I give just an example here. The symbols here, the font ensemble symbols correspond to if I activate a flow, so a flow is bin B, or if I deactivate. Linear constraints equal 1 if activate, 0 if not. I modify just the constant, so B is able to update the caches in the already explored branch and bound tree, and so it is faster for the next resolution. Okay. We can also use some tricky things like reducing the combinatorial when we are searching for fully circular plasmids. You have permutation of the solutions that represent the same thing, especially when it is circular. And so one of the solutions you can take is to have this type of thing, like the source arc and the sink arc replace one arc in the circularity, solution 1 and solution 2. And what we can do is forcing to use all the fully circular arcs, but you have to put 0 flow on the source arc and the sink arc, and you have to modify plasmid flow for that. It's not a problem, but okay. And you have also this modification. You can ensure some other performance purposes through the modification of the constants. Okay, I will not go further more into the details here. What are the objective function here? We want to find a set of a known number of plasmids that explain the best our data. So we want to maximize an explanation score, which is a sum between plasmidness score and all positive plasmidness coverage penalty. Okay, it's a long term, but it's not so complicated. Okay, the plasmidness score. We want to encourage or penalize coverage usage for contigs with a high-low plasmidness score. The plasmidness is rho I here. Okay, I is a contig, the length of the contig, and the sum of the input flow for the bin B, for the flow B of contig I. So what is it? The sum is the usage of the coverage of contig I in the bin B. Okay. More we are using coverages, more this sum is I, and if the plasmidness here, okay, is between -1 and 1. So if it's not a plasmid, it's -1. If it's a plasmid, it's 1. So you want to encourage or penalize either the plasmidness score is high, and you are robust according to the contig length. Okay. And you have this term, which is a penalty. We want to penalize the unused coverages for the contigs likely to be plasmids. So we are summing up the contigs with a plasmidness greater than 0, so likely to be plasmid contigs. And okay, this is the unused coverages. I want to present you some preliminary results on subsamples. So I tested a lot of things on the same 10 samples, bacteria short-read samples from NCBI, where we have plasmid references. We made the assembly with, so Aniket and Cedric made the assemblies with unicycler. They just filter the contigs less than 100 base pair. It's a common filtering step. They did the classification with plasmid. I did, sorry, the classification with plasmid CC, but we can also, they have also made the classification with plasgraph 2. I choose plasmid CC because it's a default classification step for G plus CC, and we want to compare with the main competitor working on assembly graphs, G plus CC. And the seeds are provided by a classification tool, Platon, with high precision but at a lower recall. But this is the purpose for the seeds. Okay. What about the evaluation? It's not an easy part because we cannot use directly the well-known in the clustering community homogeneity and completeness clustering V measures because they fail to correctly evaluate the binning, because in the hard clustering remind that we want to cluster each contig to exactly one group. This is not the case here. A contig can be shared between different groups, and we cannot adapt directly the fuzzy V measures because we're only interested in evaluating the plasmid bins, not the remain contigs. And you have a really large set of remaining contigs from the chromosome or for the viruses. Okay, it can bias a lot the statistics. So I'm using here the recall and precision measures given by PlasEval, made by the same team as Plasminflow. And so these measures are suited for evaluating the binning results. I'm now running on large band path, but okay, let's see some of the results. You have the naive approach. The naive approach is one bin containing the contigs with the high plasmid score, and that's all. Just one bin with plasmid score greater than 0. You have the hierarchical plasmid approach in the second position and third position, and you have G plus CC. Why we have duplicate the hierarchical multi-flow binning approach results here? Because in G plus CC, they connect the contigs together to form a bin, and some of the contigs were not identified as plasmid before by a classification step, here a plasmid CC, and they remove them from the final results, so they filter. We did the same in a second step. So here the first yellow violin plot is the results with contigs that can be considered as plasmid in a classification step, but they allow us to rebuild circular plasmids. And in the second step, we just remove these contigs. So we can compare to G plus CC. In recall, we are, okay, remind that it's only on 10 samples. Okay, in recall, it seems to be great. In precision, we are not so good for many reasons. I will explain better after. In F1 score, which is the recall and taking into account both the recall and the precision, we are quite in the same values, let's say. Okay, this is according to the number. Okay, G plus CC was not able to solve all the problem. One problem on 10 was not solved. I don't know really why. And okay, the figure B represents the differences of the number of bins comparing to the number of plasmids we wanted. And Okay, G plus CC is quite great because the mean is at 0 and it's not so large, the boiling plot. Okay, we are comparable. So we have to do larger benchmarks. As some conclusions, and I will interpret some results. What was our goal? Our goal is to find a set of an unknown number of plasmids, beans, that explains the best our data, the coverage, the plasmidness, the seeds, and sometimes the GC content. I based the presented work on plasmid flow, which is an iterative flow-based binning approach, ensuring that each bean contains a seed and they label the bean with GC content interval. I proposed a hierarchical multiflow approach, which searches for multiple beans in ounce, with focuses on circular plasmids, otherwise partially circular, first with seeds, and then without any obligation of containing a seed. We model connected flow with constraints directly in the MILP, and we add some performance purposes constraints. I want to send you a message for people using mixed integer linear programming. Beware of preconceptions. Okay, in general, adding new variables, adding new dimensions is really costly. And you can say, oh, I will not take the risk to do a better theoretical model because it will be too costly. Beware of preconception, because I hope I have demonstrated you some ideas that when you add new variables, but you add a new semantic, you can help the branch and bound to solve the problem, and you can to get better results. So try it, be crazy, try it, and you will see. Okay, and you can use some smart constraints to reduce the performance issues, the time costs. Okay, as some perspectives. We want, of course, to evaluate the current approach on a larger benchmark, because 10 is not sufficient. We want to continue the multi-flow binning logic, because this is just the beginning. We want to search for both circular and linear plasmids. I was just focusing on circularity. I don't want to use seeds. I mean, I don't find really relevant the use of seeds. I want to test without seeds. Less data we have, better it is. This is my philosophy. Still, there is a tricky question. Okay, we want, in the perfect world, we want one bean equals to one plasmid, a complete plasmid. But we will not achieve that. And perhaps it's better to keep merged beans and maybe splitting them after that. And how can we split them? For example, with adding typing constraints. So to add typing constraints, we have to translate the typing schemes to mathematical constraints. Then we can first make the binning, keeping merged some beans. We cannot really separate because they are too closely related. Okay, and with these new constraints, we can perhaps separate them. Again, you have to clearly describe the properties you ensure to a user. So you can say, okay, I will keep merge the beans, and I can even give you some advice on, okay, this beans is likely to be a merge of plasmids, and try to do other methods to separate them. But you have to be clear on the results you are giving. Okay. Another problem is addressing the evaluation of binning results, because the V measure and the adapted fuzzy V measure for the cluster from the clustering field are not suited to evaluate binning results. Okay. That's all. Thank you for listening. Simone.
Thank you very much, Victor, for this great seminar. If you have any question, please feel free to send an email or a message to Victor. We will put his website with all of his information on how to reach him online, and you can write him both for a question about this or for all the others activities that Victor is involved, especially in open and fairness of science. Maybe if you want to say something about those initiatives as well, you can.
Yes, I haven't prepared any slides, but in a few words. So, in fairness, we have issues on publication systems. It is well known we are facing paper mills, so spamming of papers, sometimes of bad qualities. I want to send a message. The aim of science is not spamming papers. But I know why we are spamming papers. We are spamming because we need to publish, because we are evaluated according to the number, for example, of publications. We are scientists. We decide ethically what is a good science. It takes time. We have to be sure that what we present is well presented. I'm not saying that what I have given now is well presented. I try my best, but okay. I have to take the time, be careful what I'm putting on internet, etc. It is the same for publications. So we have to redefine ethically the publication system. And especially now with IA, we will see a lot of worse than spamming, because IA will create contents and it will be more and more difficult to new scientists to explore the sciences community. And it's really important to have really good introductions into a field if you want to give new ideas. So, okay, I will stop on that and just tell you that if you are interested in publication system, you can hear on YouTube presentation of Marie Farge, which is mathematician and on quantum wavelength, and she was involved at the European Union scale about the publication system, diamond access, etc. Okay, and for ethical in general, if you are interested in the subject and in the concept of academic freedom, there are a lot of groups worldwide of researchers and teachers involved in the defense of academic freedom. We see with Trump in America, but not only in America, even in Europe, a lot of new threats on academic freedom. So academic freedom, what is it? It is our ability to work properly and ethically in science, which is really important to ensure the quality of the result and the quality of the knowledge we are giving to not only our communities, but also to the citizens. We are citizens, of course, but we are producing knowledge and we have to ensure the quality of this knowledge. So being independent of economical, political, religious powers. If you are interested, you can see the French Association for Academic Freedom, ALIA, but you can also find in your own country, perhaps, some groups defending this, and we can work together in an international working group.
Okay, thank you very much, and thank you all for following along, and see you in the next webinar. Thank you.
Thank you a lot, Simone.